Hello everyone. Today we'll be discussing about fish. Fish is nothing but fluorescent in situ hybridization. This involves hybridizing your probe that is complementary to your DNA, and this probe is attached to your fluoropores, and this is observed under the microscope. So what do you have here? You have a primary requirement of a probe that is complementary, and this probe is further attached with a fluoropore. That is what you have. Now, there are three variants of fish here. The first one is metaphase fish. The next one is the interphase or the prophase fish. The next one is the interphase or the prophase fish. Next one is interphase or prophase fish. And the last one is the fiber fish. Last one is fiber fish. See, basically, when we were discussing optical mapping in the previous lecture, optical mapping is restricted to chromosome lengths of only 1 MB. That is what it was standardized for. Standardized by using backclones, which are around 1 MB in length. That is what is optical mapping. So here in fish, you can use the entire chromosome, and all across the chromosome, you can hybridize probes that are complementary to your DNA, and then have the genome mapping done. So the first method, which includes metaphase chromosomes, just because in the metaphase stage, you have your chromosomes to be highly condensed. And you see your chromosomes as two chromatids just because the cell has already gone through the synthesis phase. So you have your chromatid here. And now let us say, if you are taking an inset of this, let us say you're taking an inset of this, so you'll have a double-stranded molecule. Entire double-stranded molecule. This double-stranded molecule is completely complementary to one another. And this runs from 5' to 3'. And this runs from 3' to 5'. Right? This double-stranded molecule is now denatured. This double-stranded molecule is now denatured. This denatured double-stranded molecule now will be like this. Let us say it's partially denatured. So we have a double-stranded molecule like this. And to this double-stranded molecule, you will now hybridize your probe of interest. You'll now hybridize your probe of interest. Now you take a probe. This probe binds to this region, which is completely complementary to the probe. And this runs in this direction. And this probe is now attached to a fluoropore, which is now observed under the microscope. This is the basic technique of your metaphase fish. However, there is a big limitation here because on a chromosome, using metaphase fish, the maximum resolution is only 1 MB, means only two probes that are at a distance of 1 MB can be clearly seen. Now, to overcome this, people thought like we should have chromosomes in extended form. So they thought that they are mechanically extended in the prophase stage or in the interphase stage. So they took prophase chromosomes or the interphase chromosomes and did fish in a similar fashion like what I have explained at the top. And here they reduced the distance and they got a resolution of around 25 kb. Now you can see that the resolution is around 25 kb, means two probes that are within a distance of 25 kb can be clearly seen. However, people still wanted a better resolution, so they went in for a fiber fish. The third one, which is called as fiber fish. Fiberfish is nothing but a modified version of optical mapping. It is a modified version of optical mapping. You already know what optical mapping is. If you don't know it, there is a separate video on optical mapping. You can go into that and understand what optical mapping is. Now, when you look at the modified version of the optical mapping, here, what do? The technique is pretty straightforward. You extend your DNA as a thread, and to this extended DNA, you would like to attach your probe. And by doing so, what happens? The resolution would definitely improve. So this extension that they achieved was through microfluidics, like what I have explained in Sapphire in the previous talk of optical mapping, microfluidics. So your DNA is in the form of a straight thread to which you have the complementary DNA. And this complementary DNA probe that is attached to the straight thread is now visualized under the microscope or can be automated like what we have seen earlier. The next important thing that we should understand here is how can we improve the stability of the probe binding? The stability of the probe is quite important because the probe that is complementary to a DNA will have competition from its complementary strand. What do I mean by this? You have your DNA, right? That is denatured, the one that we have drawn before. This denatured DNA, you have your probe that is bound. This binding of the probe has competition mainly from the complementary strand. This is template. This is a template. You have a complementary strand. Now, this complementary strand will compete with your probe to renature the template. And your probe sometimes would prevent it to bind to your template. So probes are normally prepared with PNA, peptide nucleic acids. Now, what is this PNA? Peptide nucleic acids are different from the normal nucleic acids in having a different backbone. In the normal DNA, the backbone that you have here, you can see a sugar and a phosphate backbone. Whereas in peptide nucleic acids, you would have an amide backbone. Having got an amide backbone, they'll be now having a different kind of hydrogen bonding in addition to the normal hydrogen bonding. Now, what is this? The normal hydrogen bonding between A and P, you'll have 2 covalent bonds. And simply between G and C, you'll have 3 covalent bonds. Now here, just because you have amide bonds, you have a different kind of hydrogen bonding that is there in the case of PNAs. The figure on the top here, it shows the normal hydrogen bonding between G and C. You have 3 hydrogen bonds, 1, 2, and 3, and you have 2 hydrogen bonds that are 1 and 2 here between adenine and thymine. Whereas, in addition to this normal binding, listen to me very carefully, PNAs normally will have also the normal hydrogen bonding, like what you see between A and T and also G and C, right? In addition to the normal hydrogen bonding, there will be hydrogen bonding at different positions between the guanine and the cytosine, like you see over here. There are 2 additional hydrogen bonds in addition to the 1, 2, and 3 that are seen in Watson and Crick hydrogen bonding. And totally you have 5 different kinds of hydrogen bonding that can be seen. And similarly here, between adenine and thymine, you'll have 2 other hydrogen bonds that can be formed at different positions just because of the amide backbone. And this, in effect, like you'll be having 4 hydrogen bonds between adenine and thymine. So the stability now improves just because of the increased number of hydrogen bonds, and the probes are said to be more stable. And this stability would further be enhanced mainly because of, like when you see a template, right? This template is negatively charged and this template is negatively charged. Just because both these templates are negatively charged, there's a slight repulsion between these two. And now, when you don't have a phosphate backbone, and just because you have an amide backbone, you don't have a negative charge on the probe. And just because of these two factors, what are the two factors? One, increased number of hydrogen bonds. Increased number of hydrogen bonds. That's the first factor which improves the stability. And what is the second factor? The second factor is just because there is no phosphate backbone, there is no negative charge, and there's no repulsion between the strands. And there's a slight repulsion between the complementary strand and the template strand, mainly because there is negative charge here on the template strand and also on the complementary strand. So the probe now is more stable than the normal complementary strand that can probably compete with it to renature and bind to its template. This kind of base pairing that involves other groups of purines and pyrimidines to have extra hydrogen bonds is called as Hoogsteen base pairing. Hoogsteen base pairing.